structured this talk kind of very basic for non-biology and non-chemistry people. But if that not exists, feel free to question me anything about fluorescence spectroscopy and diagnostics, even things that are beyond of what I'm going to talk about in the first few minutes of the presentation. So we all know diagnostics. How do I change? Okay. So when you talk about diagnosis, essentially the paradigm or the most universal paradigm for diagnosis is your ability to measure something. Because without an appropriate measurement scheme or method, you really can't diagnose. You don't know if something is going right or something is going wrong. And if you measure something in a biological system, then the response or the parameters that you are going to measure is going to be biological, chemical, or physical. Now, if you take these three kinds of signals that emerge out of a system, they get into the typical five senses that biological systems have: is touch, sound, light, smell, and taste. Unfortunately, the sensing systems that we use in devices are still not evolved enough to measure smell and taste. Of course, the high-end technologies like mass spectrometry and few sensors, VOC sensors, do something which is similar to smell and taste, but yet uh, they are not so good. So most often you rely on touch sensors, which means you are capable of measuring pressure of some kind. And a typical example of that is a weighing balance. It measures the pressure exerted by an object that is put over the device, and that pressure gets converted into a signal that you measure, and that correlates with the weight of the object. The second kind of signal that we very often develop into any kind of diagnosis system is sound. And the typical or the most often used technology is ultrasound, which you use not to measure sound but to do imaging. Okay. And the third, the most popular, which kind of accounts for almost 90% of everything that we measure, is light. Almost all of biology is based on the capability of measuring light. Now, when I say that, light cannot be measured directly because we really do not have mechanisms of measuring directly. So, and any sensing system that we have these days relies almost exclusively some kind of an electricity to be measured. So it can be the voltage difference that is generated out of the signal or the current that flows out of the sensor, and that is what you actually measure. Now, from light to get converted to electricity, you need to transduce. So you transduce it via something. I put in a box of direct is where I mean that you measure light directly using something called as photosensors of various kinds. So you have photodiodes, you have phototransistors, you have photoresistors, which can be used to measure light directly. So so let me get back. So we had transducers that again convert light into some form of electricity. Now, I put in a small box here as relevance versus capability, primarily because what you want to measure might have huge amount of relevance in your diagnostic capability, but yet you cannot measure it simply because you do not have the capability to measure it. For example, a particular biomarker may be highly relevant to you, but you cannot just measure a biomarker straight away. Say, for example, if you have to measure glucose straight away, there is no real sensor for glucose available. No matter what you do, you have to convert the signal from the sensor or from the glucose to get converted into either light or sound or touch, pass it through a transducer element, convert it to an electrical signal, and now we are capable of measuring it, with reliable precision and accuracy. So we'll focus more about light and how light dominates most of the diagnostics that we do these days. And before I go to fluorescence, we know the typical property that is most often used for doing any kind of measurement is absorbance. Absorbance means the chemical nature of the molecule allows certain wavelengths of light to enter the molecule but not exit the molecule. That means the light is absorbed. Now, typically based on the molecular nature of that particular analyte, it will have different energy structures that will correspond to different wavelength of light that can be absorbed. So something can absorb only in the blue, something can absorb only in the violet, something can absorb in the green, yellow, red, and some molecules can have a much wider spectrum of light that they can absorb. Some molecules can work only in the UV, some molecules can work in the IR, and henceforth. So almost all techniques that are based on absorbance follow the simple principle of particular component of the molecule that can be absorbed or a particular component of the molecule absorbs a light of a certain frequency or the wavelength. Okay. So now, if this particular molecule that absorbs light be used as a sensor for transducing the light signal or transducing anything, then absorbance molecules, there's only two ways this molecule can behave in response to your sensing activity. The first is there'll be a change in intensity. That means compared to a resting state of spectral feature, which is composed of the wavelength and the intensity at a particular wavelength, the intensity can go up or it can go down. Conversely, the intensity may not get affected, but the peak wavelength where the molecule absorbs may get affected due to the molecule or the behavior or the sensing capability being done by the molecule. So these are the two essential behaviors that you can expect out of this system. Now, if you see, this has been very widely utilized in almost everything that biologists and chemists do for measurement, one of the most fundamental. But for an example, a very simple urine analysis strip, which contains pads, and you dip it in urine, and there are so many different parameters of the urine that can be measured by this strip. And what you see in all these cases, there is a shift in the color, which means there is a shift in the wavelength that the light is absorbing. And hence, the whole act of sensing is happening. However, it is widely known or very well accepted that absorbance based methods have low sensitivity and low specificity. Now, if somebody can remind me at the end of the talk, we can go back to why we say that it has low sensitivity and specificity. The other behavior where light can be used to do the sensing is looking at the property of fluorescence. Now, fluorescence is different from absorbance in being that my molecule is a magical molecule where I throw a particular wavelength of light on it. So it absorbs a particular wavelength of light. Of course, that wavelength of light is absorbed by that molecule, just like absorbance. But now this molecule starts emitting another wavelength out of the system. So if I throw UV light onto the system, I may start getting a blue or a green light coming out of the system. Now this happens because these molecules, the electronic transitions in these molecules are slightly different, where the electron, after getting excited from the X0 state to the S1 state, now has a path to come back to the ground state. And in the process of coming back, it releases the energy. And since the released energy is lesser than the energy it absorbed, so lesser energy means higher lesser frequency or higher wavelength. So you always see a shift between the light that you throw into the system and the light that comes out back out of the system. Now, interestingly, for these kinds of molecules, the local environment plays a very crucial role in determining how much of the light comes out. And just like absorbance, this local environment can dictate whether the light is in the wavelength of the light coming out is retained or not, and as well as the intensity goes up or goes down with respect to the signal. Now, fluorescence is nothing magical. It has been there all along. And so fluorescent molecules are almost there all along, and they're used in various kinds of applications. The first image that I show is a UV photography of a vitiligo patient, the hand. You see the areas that have reduced pigmentation fluoresce very brightly versus the areas that have rich amount of melanin. So, and this color, this is called as autofluorescence. That means you have not put any marker into the system. The biological system has molecules generated within it which show this property of fluorescence. So you throw UV light and you start getting blue light out of the system. All insect species, and in fact almost everything that is living, has some or the other kind of pigment generation to protect itself. So the second image is a huntsman spider, where you throw UV light and you see pigments of green and blue color coming out. The third image is more interesting, is a flower. If you see, the whole flower looks yellow in white light, whereas if you look at it in the UV light, the edges of the petal fluoresce blue, fluoresce yellow, while rest of the flower just absorbs. So there's a unique distribution of chromogenic pigments which are visible only in white light versus fluorogenic pigments which are visible only in UV light. These fluorescent pigments are being used widely in fashion these days. You see a lot of pubs and discos being coated with fluorescent colors on the walls. People are wearing makeups with fluorescent colors so that they look different when UV light is projected on them. Fluorescence is also used since a very long time to look at for doing water tracing, which means that when you are unaware of how the water streams connect underground, and or you'll want to look at the currents that are happening in water, so you start putting huge amounts of fluorescent molecules into a water stream and try to look where else you start getting the fluorescence pigments out in different water streams. So these have been used to discover hidden lakes, hidden water bodies, hidden connections between streams and rivers, and lots of such activities. This is another application where the diagnostic relevance of fluorescence comes in. So if you are doing fundoscopy to look at the retina's health in various cases, it's a very commonly used dye which is called as indocyan in green and fluoresces in far red. So it's beyond red and slightly before the infrared. So in the first image, you see that upon the veins that carry or the blood arteries that carry the dye fluoresce very brightly. So you see a huge amount of contrast in the image, whereas after some time when all the ICG is metabolized and it is reduced in circulation, you see those details are completely lost. So it is used as fluorescence has been very widely used as an enhancer for imaging. Another application, again very commonly used in personalized medicine, is fluorescent in situ hybridization, where you are trying to look at gene additions or gene duplications, not at the molecular level, but using a molecular technique, but looking at the chromosomal level. So we know that a lot of genetic disorders happen due to either a deletion of a gene or an addition of a gene or a duplication of a gene or something or the other. How do we know that a particular phenotype that we are looking at is due to a certain a deletion or addition? Herein, what you do is you take a nucleic acid sequence and label it with a fluorophore directly or indirectly, and try to give it into a preparation of chromosomes. Now, if this goes and binds to its complementary DNA strand within the chromosome, you know that this gene is present. If it does not, you know that the gene is absent. So, and there are various formats of fish by which various kinds of diagnostic utility has been derived out of it in personalized medicine. The next general application is the real-time polymerase chain reaction. I put it for people who are not from a biology background, and especially with the COVID news all over. It's this particular technique has become a hero in this whole war. So here, what happens is the idea is to look for a gene, whether it is present or not. In this case, you take the sample from a human, suspected infected human, and you want to look if the RNA of the virus exists in the sample. So if the RNA exists, you know that yes, this person is infected with the COVID, and if not, fine, you are safe, you are happy. But how do you do that? Because the amount of virus is going to be extremely low. And secondly, it's RNA. Humans also contain RNA. So how should we be able to do this, bifurcation of presence or absence? Now, this is done by a modified version of the polymerase chain reaction. And polymerase chain reaction does something very simple. So the whole genome or the DNAs consists of a huge number of long length of nucleic acids. And say, for example, you know that this particular sequence of DNA is unique signature of, say, COVID, then I have to hunt for that sequence. And how do I hunt for that sequence? I create a strand synthetically, which is an opposite letter of, or the complementary letter of that particular strand. Now, by default, the double helix base pairing rule, two strands will complementarily bind to each other. So I have a recognition element, but my DNA quantity is extremely low. So I will not have enough capability to detect it. So what do I do? So I need to amplify. So I do this process of amplification by the first stage of binding and the second stage of chain elongation, or the extension as given here, where more nucleic acids complementary to the strand existing start building on a chain. So I start with, say, one copy of the viral RNA and one sequence of primer, which was kind of my target molecule. And now I built two sequences. I repeat the process again and again. And by the factor of 2n, after a couple of cycles, 30 cycles or so, I would end up having a crore molecules of the exact sequence. And now at this particular number of molecule present, I can put in a probe which binds there, and that probe tells me, yes, this sequence is present. So if you look on the right side of the right side graph, you start with very low copy numbers, and as you increase the cycle number of the PCR reaction, your fluorescence starts going up. Now, the curve, the slope of the curve, and the place where the curve starts to go up is indicative of how many initial copy numbers were existing. So if you had 0, then your whole amplification cycle will be 0 into 2 to the power n. If you had 1 copy, your total amplification will be 1 into 2 to the power n, and so on. So if you have very high levels of infection, that means you have very high levels of viral RNA. You do this process and you will have huge amount of fluorescence coming out of your system at the end of the cycles. And you know that, yes, I am infected with COVID. Now that was more at the DNA and RNA level. Now, almost 80% of all molecular diagnostics which is done is by looking at the level or the presence of a particular protein that we are interested to look at. So the hypothesis being that if my protein is present, yes, I may have the disease, or if my protein is absent, then I may have a disease, depending on the case specific. For example, a cell becomes a cancerous cell only when there are certain proteins expressed on its surface. Then you know that this is a cancerous cell and not a normal cell. Similarly, if you have a chikungunya infection or a dengue infection, you will come to know of that only if you have the antibodies to the dengue virus available in your blood. And all these is done by a technique called as ELISA, which is enzyme linked immunosorbent assay. So what happens here is you you have a molecule that you want to look at, depicted in yellow here, and then you will have a primary antibody. A primary antibody is a molecule that will specifically bind to the molecule of your choice. And then the primary antibody would be linked to an enzyme. The enzyme will be exposed to its substrate, and the product of that substrate or the enzyme converts that substrate into a colored compound. And based on the amount of color that is hereby formed, you know how much of your molecule of interest exists in the solution. And this can be done in three different formats popularly. One is the direct format, indirect assay, and the captures or the sandwich assay. Now here, if you replace the enzyme with a fluorescence tag, you still can do the same thing, albeit the amplification that the enzyme is bringing into the system gets lost. So here, if you convert it to a fluorescence, then you should have a very nice detection system to be able to directly measure fluorescence. The advantage here are twofold. Number one, since you are only using a fluorescent probe to do the measurement, your chances of false positives or false negatives are very, very limited. That is, only if something is bound, use the fluorescence is there, and no background amplifications happens due to the presence of an enzyme. The other advantage, which is very commonly used, is this: because of the simplicity of the format and its specificity, you can do it directly on a cell. Of course, the first method was also people used to do that on cells, fixed cells, non-living cells, uh, to look at whether where a particular protein was present. But the second method allows you, or the fluorescence method allows you to do that in a living cell, because the addition of that fluorophore really does nothing bad to the living cell. Now, when we are talking about sensing per se, now whatever I talked about until now was about what essentially is fluorescence and where you are using it. But in none of the systems, fluorescence in all those systems, fluorescence was essentially a signal generator, a point which was generating some signal which I can measure. But the fluorophore was not a molecule that was doing any sensing per se. The sensing was happening due to other components in the system. So there's one beautiful property in all colored molecules, and that is called as solvatochromism. That is, these molecules change their absorbance or fluorescence based on the local microenvironment. On the left, you can see a dye which is called as a Raycud's dye, and that the same molecule has multiply different colors the moment you put them in different mixes of hydrophobic and hydrophilic mixes. So the polarity of the system gives rise to different colors. A same behavior is seen in a different fluorescent dye here, not an absorbing dye, but a fluorescence dye, where the output fluorescence depends upon the nature of the polarity, the solvent that it is. Now, this particular behavior has been very widely utilized to do direct sensing by the fluorophore. So I'll bring in here our case study, use case study. We have been trying to develop a assay and a device that should allow us to measure urine microalbumin. Now, why that is important, you should know that our kidney's main function is to filter out urea and the waste into vast amounts of liquid and keep everything else that's in the blood intact. Now, when the kidney gets damaged, the basal membrane is unable to do this filtration very efficiently, and therefore the albumin from the blood starts leaking into the urine. And that is of diagnostic relevance because albumin should not be there in the urine, or very, very trace amounts of albumin should be there in the urine. Now, how do you measure this albumin in the urine? For the past 20 years or so, people have resorted to using antibodies against albumin, so that when so you take urine into a strip or a dipstick, and when you add the antibody to albumin, if albumin is present, your antibody binds, and you kind of do a kind of an ELISA to set up to measure the albumin. And this is the format which a lot of methods use. The other method where people slightly change this format and do immunoturbidity, that is, I have albumin in my solution, urine in this case, and I add an antibody, polyclonal antibody to it, and the polyclonal will bind many molecules of albumin, and similarly one molecule of albumin will be bound by multiple antibodies, and thereby a kind of a coagulative phenomena happens. It's not true coagulation, but it is agglutination, the same method that happens when blood grouping test is done. So if the turbidity increases, then albumin is there, and depending on the level of turbidity, you can quantify how much of albumin is there. So you see, for both these methods, my primary requirement is to have antibodies towards albumin. Now, if I want to get this test at a point of care, or a person who wants to use it at his home to measure his albumin levels, in case of patients who suffer from chronic kidney disease, um, it's very difficult to do that because antibodies need to be stored at 4 degrees centigrade. Heat shock, giving a heat shock to these antibodies are going to lose their capability of binding. And thirdly, the amount to measure agglutination or to measure an ELISA, I need really very sensitive devices that can measure those small amounts of changes. So clearly I cannot do that. So what we did at Prantae, we started to look at various kinds of molecules that will show a solvatochromic behavior upon binding to albumin. And we found one, and there are other people who have found others as well. And this molecule happens to be a fluorescent molecule, so you excite light at one wavelength, and if the molecule binds to albumin, it will start emitting at a different wavelength. And based on how much of light is getting emitted, you are capable of quantifying how much albumin is present in a very, very simple format. Now, to do that, you need a device that can measure low quantities of signal. And this is relevant for fluorescence because the amount of light that comes out of fluorescent molecules is really very low. So you should be able to, collect that light very effectively. Next, you should be able to amplify that signal. Now, typically when you amplify the signal, you also get increase in noise. So you have to reduce the noise. And more importantly, the whole device plus assay should be able to give you a quantitative response with respect to the amount concentration of the molecule that you are trying to sense. And if you want to really make it as a business case study, and you want people to buy and use it, the device should look good, it should be affordable, yet it should function. And that's too much to ask for like this. Why? Because typical fluorescence spectrometers can cost anywhere between 4.5 lakhs to 25 lakhs. Okay, and by far they are always used in research laboratories or QC centers, almost never at a consumer end. And that was a big challenge for us. I'm showing you our success where we were capable of creating this system where we could obtain the limit of detection of 2.7 milligram per liter. So you can imagine how little quantities of albumin can be measured with our system. And the system is integrated with a mobile phone to be able to do the measurement. So you see the mobile phone and the device at the back, which houses all the electronics and tests. You see the user putting the urine into a cartridge system, which is already pre-coated with the reagents. And the cartridge then goes into the device, and the device gives an output. Now, let me just tell you that this kind of an LOD is equivalent to sensitivity with what is observed using the ELISA based method for microalbumin measurement. So these are the different formats that the device is designed to be. The format on the top extreme right is a clamp-on adapter for mobile phones or smartphones, where you can use the camera of your smartphone as a measurement device, and you can do this assay. Uh, the one on the right, shaped like a small baby, is designed for pregnant females uh, who want to monitor high risk pregnancies, because there is a disorder in pregnancy called as pregnancy induced hypertension, which sometimes leads to preeclampsia. And one of the biomarkers for detecting preeclampsia is the albumin in the urine. The bottom design that you see is more for generalized usage. You, me, anybody can use. Looks good and is a small device. So with this, I kind of end a very fundamental, or rather a very brush through about how fluorescence can be utilized for developing diagnostics that can go to the user end. And I'll be open for questions.